Welcome, everyone. We're diving deep into the world of genetic engineering today. I mean, it's pretty mind-blowing to think we can actually tinker with the very essence of life DNA.
It is remarkable.
It really is. Now, for this deep dive, we've got some seriously fascinating material to unpack. We're going to be looking at Jennifer Doudna's book, A Crack in Creation. She's a Nobel laureate and a pioneer in CRISPR.
A fantastic read.
Absolutely. And we'll also be drawing from a whole bunch of scientific publications to really give you the full picture, the history, the how it works, the amazing possibilities, everything. But we want to go beyond just the hype you know.
Right.
Our mission is to equip you with a deep understanding of genetic engineering.
The good, the bad, and the ethically complex.
Exactly. So before we get to the CRISPR craze, which seems to be all over the news these days, let's rewind a bit. What can you tell us about those first attempts at gene editing, like before CRISPR was even a thing?
Well, way before we could actually edit genes with any precision, scientists needed to figure out how they even worked and how to change them.
Right, the basics.
Yeah. And there was this fascinating case early on, Kim's case of WIN syndrome. Now, this involved spontaneous chromosome shattering, which incredibly led to a cure. It was like a glimpse into the body's own potential for self-repair at the genetic level.
Wow. So the body kind of hit the genetic reset button.
In a way, yes. But as you can imagine, that sort of thing is incredibly rare.
Right. Not something you can rely on.
Exactly. So scientists knew they needed to find more controlled methods. And those early gene therapy attempts, they used viruses, specifically viral vectors, to deliver therapeutic genes into cells. But it came with challenges too. I mean, there was that tragic trial back in '99 where a patient died because of an immune response.
Oh, right. I remember hearing about that. So it's kind of this delicate balancing act using the power of viruses while making sure they don't backfire.
Exactly. And then scientists turned their attention to a process that happens naturally in our cells. Homologous recombination. Think of it like a DNA swap meet,
Okay, I like that analogy.
where similar sequences are exchanged or repaired. And this laid the groundwork for more targeted gene editing techniques.
Ah, so it's kind of like finding that perfect puzzle piece.
Precisely. And a major breakthrough came with Kopecky, Smithies, and Evans. They showed that this homologous recombination thing, it worked in mice. It was a huge deal. They even won the Nobel Prize in 2007.
So that was a massive step forward. But from what I understand, even with that breakthrough, there was still a need for something even more precise, like a tool that could zero in on a very specific spot in that vast, expansive DNA.
You're absolutely right. And that's where designer nucleases come in. These were engineered proteins built specifically to target specific DNA sequences. One of the early examples was something called zinc finger nucleases, or ZFNs for short.
Zinc fingers.
Yeah.
That sounds more like something out of a fantasy novel than a science lab.
It does have a certain ring to it, but these finger-like protein structures, they can actually recognize specific DNA sequences. Designing them, though, was a complex process.
I can imagine.
Then along came talons, which were easier to engineer, making gene editing more accessible.
Okay, so we've gone from relying on super rare natural events to actually creating designer proteins that can target specific genes. That's a pretty amazing leap.
It is. But even with all that progress, there was still a need for a tool that was more efficient, more adaptable, and easier to use. And that's where CRISPR comes in. It really revolutionized the field.
CRISPR, the rock star of gene editing. But before we dive into the nitty-gritty of CRISPR, I'm curious about its origin story. Jennifer Doudna's first encounter with CRISPR, it was quite serendipitous, wasn't it?
Oh, it absolutely was. It all started with a casual chat with a colleague about this, well, kind of strange bacterial defense system.
A bacterial defense system. That doesn't sound like the most obvious starting point for a gene editing revolution.
It doesn't, does it? But it turns out bacteria have this incredible way of fighting off viruses. And the system they use, it's called CRISPR. And let me tell you, it's ingenious.
Okay, so how does this bacterial defense system actually work? It sounds a bit like science fiction.
Imagine this. It's like a bacterial immune system that keeps a record of past viral infections. So they capture little snippets of viral DNA, and then they create RNA guides from them. These guides, they're like genetic most wanted posters.
Interesting.
That allows the bacteria to target and destroy any matching viral DNA that tries to invade again in the future.
So it's like the bacteria actually have a genetic memory of their enemies.
Exactly. And the protein responsible for this DNA cutting, it's called Cas9.
Cas9, the molecular scissors that make CRISPR so powerful. But I understand it wasn't immediately obvious that this bacterial defense system could be used for gene editing in other organisms.
You're right. It took some brilliant minds to figure that out. And that's where Doudna and Emmanuel Charpentier come in. They did some groundbreaking work showing that CRISPR-Cas9 could be programmed to cut DNA at specific locations, not just in bacteria, but in any organism.
So their work was a real game changer.
Oh, absolutely. Their 2012 paper, it was like opening a floodgate. Suddenly, scientists had this incredibly precise, efficient, and versatile tool at their disposal.
And how did the scientific community react?
With incredible enthusiasm. I mean, within a very short time, CRISPR was being used to edit genes in all sorts of organisms, you name it, bacteria, plants, animals, you name it. It was incredible.
It's amazing how quickly this technology took off. And as we'll explore further in this deep dive, CRISPR goes way beyond just the lab. It's having a huge impact on agriculture, medicine, even conservation.
It's truly transformative. I mean, it has the potential to reshape our world in some really profound ways. It really is. And what's fascinating is how CRISPR's applications are not just limited to research labs. We're seeing its impact in agriculture, too. It's being used to enhance crops in some pretty incredible ways.
Yeah, disease-resistant rice, mildew-resistant wheat. I even read about mushrooms that don't brown. It's mind-blowing. And what I find really interesting is that unlike traditional GMOs where you introduce foreign genes, CRISPR just makes precise edits to the organism's own DNA.
That's a key distinction. And it often fuels a lot of the debate around genetically modified foods.
For sure. I remember reading about the aquadvantage salmon,
Yeah.
the genetically engineered fish that grows faster, and a pretty bumpy road to market, didn't it?
It definitely did. It sparked the whole Frankenfish debate, as some called it. It just goes to show how complex public perception can be and all the regulatory hurdles around genetically modified organisms, even when you're not introducing foreign genes.
It's like CRISPR has kind of blurred the lines in the whole GMO debate, hasn't it? It makes you question what we even consider natural versus engineered when it comes to our food.
It has, absolutely. And moving beyond the dinner table, CRISPR is also making waves in animal breeding.
Oh yeah, the double muscling thing, right? Achieved by editing the myostatin gene. You see it naturally in some cattle breeds like Belgian Blue, but now they're replicating it in other animals, pigs, even dogs.
Exactly. And it goes beyond just muscle mass. CRISPR could be used to create disease-resistant livestock, too. Pigs immune to porcine reproductive and respiratory syndrome virus, for example. That's a huge problem in the swine industry.
It would be a game changer, for sure, both for the farmers and for animal welfare.
Absolutely. And the possibilities don't stop there. Micropigs, humanized pigs for organ transplantation. Some researchers are even talking about the potential for reviving extinct species.
Woolly mammoths, here we come. It's starting to sound a bit like Jurassic Park.
It does, doesn't it? But as awe-inspiring as all these applications are, there's another one that's potentially even more revolutionary and maybe even a bit concerning, and that's gene drives.
Gene drives. I've heard the term, but I'll admit I'm not entirely sure what it means.
Think of it like a genetic chain reaction. It's a gene that can spread through a population way faster than normal inheritance would allow. And CRISPR has made this concept a reality.
So instead of a gene being passed down to, say, 50% of offspring, a gene drive could make sure it's passed down to almost all of them.
Precisely. And that has huge implications, especially for controlling populations of disease-carrying organisms like mosquitoes. Imagine potentially wiping out malaria by releasing gene-edited mosquitoes that can't transmit the parasite.
That would be incredible. A total game changer for public health. But I imagine there must be some pretty big risks involved, too.
There are, for sure. Concerns about unintended consequences, the potential for misuse. Those are very real. What if a gene drive ends up affecting other species? What if it's used for harmful purposes, like, creating some kind of dangerous gene drive?
It's like opening Pandora's box, isn't there? The potential for good is huge, but so is the potential for unintended consequences.
Exactly. And that's why it's so important to move forward cautiously, to have open and transparent discussions about the risks and benefits, and to have really robust ethical guidelines in place for how we develop and use these gene drives.
It's a lot to consider, but I think it's time we shift our focus from mosquitoes to humans. Let's talk about CRISPR's impact on human health.
A fascinating area, no doubt, full of potential and challenges.
Absolutely.
Yeah.
From what I read, CRISPR holds incredible promise for treating and even curing genetic diseases.
Yes, definitely. We've seen amazing success in animal models. CRISPR has been used to cure things like muscular dystrophy and certain metabolic disorders. And in human cells, it's been used to fix mutations that are linked to a whole range of devastating diseases.
It sounds almost miraculous. But how does this actually work when it comes to treating people?
Well, there are two main approaches. There's ex vivo gene editing and in vivo gene editing. Ex vivo means modifying cells outside the body and then putting them back in.
So ex vivo is like taking the cells out for a tune-up and then putting them back in.
That's a great way to put it. And this approach has been showing a lot of promise for blood disorders like sickle cell disease and beta thalassemia. They can take the patient's own stem cells, edit them, and then transplant them back.
And what about in vivo? Is that more complicated?
It is, definitely. One of the biggest challenges is getting CRISPR to the right tissues in the body. And then there's the whole issue of avoiding immune responses, making sure the CRISPR components stay stable inside the body.
So how are scientists tackling this delivery problem?
Well, they're trying all sorts of different strategies. One is using viral vectors, things like adeno-associated viruses or AAVs. They can be really effective at delivering CRISPR into cells.
Wait, AAVs. Those sound familiar. Didn't we talk about them earlier with the early gene therapy trials?
We did. They've been used in gene therapy for a while now, and they're proving to be really valuable for delivering CRISPR, too. But scientists are also working on non-viral delivery systems like lipid nanoparticles. They're basically tiny packages that can carry CRISPR into the body and release it in a controlled way.
It's incredible to think about all the precision involved in all this.
It really is. And while there are still hurdles to overcome, in vivo gene editing has enormous potential. It could be used to treat a whole range of diseases.
And there have already been some pretty remarkable successes, haven't there?
Absolutely. There's the inspiring story of Layla Richards. She had leukemia and was treated using gene-edited T cells. It was a major milestone, you know? It really showed the world just how powerful gene editing can be.
That's amazing. And it highlights another area where CRISPR is making a big impact: cancer research and immunotherapy.
Exactly. CRISPR is helping us to better understand how cancer works, and it's paving the way for more effective and targeted cancer treatments.
It's incredible. Like, we're moving towards a future where we could potentially reprogram our own immune systems to fight cancer.
It's exciting. But as we dive deeper into the potential of CRISPR, we also need to address the ethical considerations, especially when it comes to editing the human germline, making changes that would be passed down to future generations. This is where things get really complex.
It does get complicated. I mean, the idea of editing the human germline,
Hmm.
it raises so many questions. It could potentially eliminate inherited diseases, but then there's the whole designer babies concern. And what about all those unintended consequences for future generations?
Exactly. And as Doudna points out in her book, these concerns aren't just hypothetical. She talks about this biotech CEO who basically envisioned offering CRISPR babies with customized DNA mutations to eliminate the risk of genetic diseases.
It sounds like something out of a sci-fi movie. But with CRISPR, it's starting to feel like a real possibility.
It is, and that raises some really big questions. Who gets to decide how this technology is used? What limits should we place on our ability to manipulate the very essence of what makes us human? Is it up to scientists, governments, individuals? These are questions that we all need to grapple with as a society.
It's almost like we're holding the future of humanity in our hands, and we need to figure out what we want that future to look like. I was reading about the Asilomar Conference back in 1975. Scientists got together to talk about the ethical implications of recombinant DNA technology. Do you think we need something like that for CRISPR?
I think that's a great point. The Asilomar Conference set a really important precedent. It showed that the scientific community has a responsibility to think about the potential consequences of their work. And Doudna herself has been very proactive in organizing meetings and workshops to bring scientists, ethicists, and policymakers together to discuss the implications of CRISPR.
So there's definitely a growing awareness that we need some clear ethical guidelines and regulations, especially for germline editing in humans. But establishing those guidelines, that's no easy task.
It's not. We need to weigh the potential benefits against the risks and the broader societal implications. And it's crucial to have a diverse range of voices in this conversation.
Absolutely. One thing that worries me is the potential for this to exacerbate existing inequalities.
Oh, for sure. Imagine a world where only the wealthy can afford to have their children's genes edited to make them healthier, stronger, smarter. It could create a whole new level of social stratification, a genetic divide.
That's a scary thought. And some disability rights advocates have raised concerns too. If we're using gene editing to fix certain traits, it could lead to a less inclusive and accepting society, one that devalues the diversity that makes us human.
That's an important perspective. The goal of gene editing should be to empower people, to enhance human well-being, not to create a society that prizes uniformity over diversity.
It's such a complex issue with no easy answers. But we can't shy away from these tough conversations. CRISPR has too much potential to ignore, but the ethical challenges are just as important.
I agree. The stakes are incredibly high. Genetic engineering has the power to reshape our world, and we have to make sure it's used responsibly and for the benefit of all humankind.
Well said. This has been a fascinating journey so far. We've covered so much ground: the history, the science, the potential, the ethical considerations. It's amazing to think that we're living in a time where we can actually manipulate the very essence of life.
It's a future full of possibility, but it's a future we need to navigate carefully, with wisdom and a deep understanding of what our choices could mean for humanity.
And to our listeners, thank you for joining us on this deep dive into the world of genetic engineering. We encourage you to keep learning, stay informed, and get involved in this conversation. The future is in our hands.
Yeah, it really gets you thinking, doesn't it? I mean, when we talk about editing genes, the very building blocks of life, it kind of makes you question what it even means to be human. It does, for sure. I mean, it's a question philosophers and theologians have pondered for centuries, and now it's coming into play in the world of science.
It's true, and as we've been talking about throughout this deep dive, the ethical considerations are huge. But we've also seen some incredible progress, right? I mean, CRISPR is already being used to develop potential cures for some devastating diseases. It's helping to enhance crops for food security. It's even advancing our understanding of cancer. It's pretty remarkable.
It is, absolutely. In her book, Doudna highlights some amazing examples of how scientists are using CRISPR to push those boundaries, And it's
Yeah.
not just those high-profile applications like curing genetic diseases. It's so versatile that it can be adapted to tackle all kinds of problems in medicine, agriculture, you name it.
We touched on the potential for agriculture earlier, and I think it's worth emphasizing just how big of an impact this could have. I mean, crops that are more resistant to diseases, pests, even herbicides, that could be a game changer for global food security, especially with the world's population growing all the time.
Absolutely. It's a crucial area of research, especially with climate change and everything. And it's not just about yields either. CRISPR could be used to make crops more nutritious.
hmm
Maybe even address those micronutrient deficiencies that affect millions of people.
It's amazing to think that we might be able to engineer crops that are not just tougher, but also healthier. That would be a huge step towards a more sustainable future.
It would be, for sure. And it's not just a fantasy. Scientists are already making progress.
That's exciting. But as we've talked about, it's important to proceed with caution too, right? Make sure we're using these tools responsibly and ethically.
Oh, absolutely. Doudna really emphasizes the need for open and honest conversations involving scientists, ethicists, policymakers, even the public. Everyone needs to be part of the conversation to make sure that genetic engineering is used in a way that benefits everyone fairly and ethically.
It's a good reminder that with scientific progress, there always needs to be that ethical piece too, and open communication. We can't just get caught up in what's possible without also considering the potential downsides. And we have to make sure it's used for good.
I couldn't agree more. The story of CRISPR, it's a testament to human ingenuity and how much we can achieve when we work together. But it also reminds us that we need to be responsible with this power. We have to stick to those ethical principles.
This deep dive has been incredible. We've covered so much: the history, the science, the potential, the ethical side of things. It's mind-blowing to think that we're living in a time where we can actually shape the very fabric of life.
It is. The future is full of possibilities, but we need to move forward thoughtfully and with a lot of wisdom. We need to understand the implications of the choices we make.
And to our listeners, thank you for joining us on this deep dive into the world of genetic engineering. We hope you've learned something new and that you'll continue to explore this fascinating topic. Keep asking questions, stay informed, and be part of the conversation. Because ultimately, the future is in our hands.